Welcome to SWAYAM NPTEL course on food microbiology for safe and sustainable food systems. We were discussing mechanism of microorganism control in food, and we have discussed thermal processing of food, and in this we have already discussed the blanching, pasteurization and appertization, and in another lecture sterilization. And now we will be discussing thermal death rate kinetics in this lecture, and we will be covering thermal death rate kinetics of microorganisms. In that, decimal reduction time, that is d value, half life period, thermal death time, that is f value, and in that 12D concept, temperature dependence of reaction rates, the Arrhenius equation, the Q10 value, and thermal resistance constant, that is Z value. The thermal death rate kinetics of microorganisms. When microorganisms are subjected to temperatures beyond their optimum, they lose their viability. Assuming that a single enzyme inactivation will inactivate the cell, and then in suspension of microorganism of a single species at a constant temperature, the death rate, that is the rate of destruction, is proportional to the number of microorganisms. That is, in differential form it is written d10 by dt, dn by dt is equal to minus kn. The above is the first order equation where n is equal to number of viable organism at a given time, t is time in minute, and k is reaction velocity constant per minute, which is a constant for a particular temperature and a type of organism. The rearranging and integrating equation 1, we can get that integrating within limit n0 to n, dn by dt is equal to minus from 0 to t k dt, then it comes ln, that natural log n0 by n, that is equal to kt, where n0 is the original number at t is equal to 0, also called the concentration level of initial concentration of microorganisms, and n is the number at time t, also called the sterility level. Also, equation 3 can be rewritten as n is equal to n0 e to the power minus kt. Now, let us solve one question. The question is, the results of a thermal resistance experiment gave a d value of 7.5 minutes at 110 degrees Celsius. If there were 4.9 into 10 to the power 4 survivors at 10 minutes, determine the ratio n for 5, 15 and 20 minutes. So, let us solve this. It is given d is equal to 7.5 minute, n is equal to 4.9 into 10 to the power 4 at 10 minutes. So, k is equal to 2.303 divided by d, that is equal to 2.303 divided by 7.5, that is equal to 0.307. Now, that n by n0, that ratio is e to the power minus kt. So, n0 is equal to n by e to the power minus kt, that is equal to 4.9 into 10 to the power 4 divided by e to the power minus 0.307 into 10, that is equal to 1.055 into 10 to the power 6. So, like for 5 minutes, for 5 minutes n by n0 is equal to e to the power minus kt, n5 is equal to n0 e to the power minus kt, that is 1.055 into 10 to the power 6 into e to the power minus 0.307 into 5, that will be equal to 2.27 into 10 to the power 5. This will be the answer. Likewise, you can calculate for 15 minute and 20 minute also. And the question number 2, that is how many logs of microbial reduction occur if a population of 10 to the power 7 is reduced to 10 to the power 3. So, log reduction is equal to how much? That log n0 minus log n, that is log 10 to the power 7 minus log 10 to the power 3, that is equal to 7 minus 3, that is 4. That is the answer. Now, decimal reduction time, that is d value. The d value which denotes the decimal reduction time is the time required at a specific temperature and under specified conditions to reduce a microbial population by 1 decimal. The decimal reduction time is dependent on the temperature, the type of microorganism and the composition of the medium containing the microorganism. It is the time required at a certain temperature to kill 90% of the organisms being studied. Hence, decimal reduction time is the time in minute during which the original number of viable microbes is reduced by 1/10 of its initial concentration. That is, it is the time necessary for 90% reduction in microbial population. When referring to d values, it is proper to give the temperature as a subscript to the d. For example, a hypothetical organism is reduced by 90% after exposure to temperature of 148.89 degrees Celsius for 2 minutes. Thus, the d value would be written as d suffix 148.89 is equal to 2 minutes. Now, substituting t by d into equation number 3, we will get n by n0 equal to 1 by 10, that is equal to e to the power minus kd, or taking logarithm on base 10 on both sides and solving for d, we get d is equal to 2.303 by k. Now, combining equation 2 and 3, we will get t is equal to d log base 10 n0 by n. This figure shows the effect on an organism held at temperature for a period of time, and time is the only variable. It started with 10,000, that is 10 to the power 4 organisms, and after 10 minutes 90% of them have been destroyed, leaving 1000 organisms, that is 10 to the power 3. This is referred to as 1 decimal log reduction. In this instance, d value is equal to 10 minutes. For every additional 10 minutes at same temperature, this process kills an additional 90% of the organisms. In the graphically, it is represented here. If the log n, that is the log number of population, is plotted against the time, then this is the d value in 10 minutes. Now, it is important to remember that the d value for an organism is significantly different at different temperatures, which can be seen in the adjacent graph. D value changes dramatically when the temperature is varied. It is easy to understand that as the temperature increases, it takes less time to destroy population of organisms. Therefore, the d value, that is the time needed to kill 90% of the population, becomes smaller as the temperature rises. Let us take example that a microbial population is reduced from 10 to the power 6 to 10 to the power 2 in 8 minutes at 121 degrees Celsius, then calculate the d value. As we know that T is equal to D is equal to T divided by log base 10 n0 by n, that is T divided by log n0 minus log n, that is equal to 8 minutes. It is given here, 8 divided by log 10 to the power 6 minus log 10 to the power 2, that is equal to 8 divided by 6 minus 2, that is equal to 2 minutes. This is the answer. And in another example, a microbial sample of 10 to the power 5 is exposed to heat with a d value of 5 minutes. How many survivors remain after 15 minutes? So, this can also be solved in that fashion only. Here n0 is given 10 to the power 5, d value is given 5 minutes, and t is also given, that is 15 minutes. So, T is equal to D log base 10 n0 by n, that is equal to or we can write in this fashion that n0 by n is equal to 10 to the power T by D. Since it is getting reduced, so negative sign. So, n is equal to n0 into 10 to the power minus T by D. So, that is equal to 10 to the power 5 into 10 to the power minus 15 divided by 5, that is equal to 10 to the power 2. So, this is the answer. In another example, a bacterial culture is subjected to thermal treatment at 70 degrees Celsius. The initial population is 10 to the power 6 CFU per ml, and after 5 minutes the population decreases to 10 to the power 3 CFU per ml, then determine the d value at 75 degrees Celsius. So, here we know that T is equal to d log n0 by n, that means d is equal to T divided by log base 10 n0 by n, that is equal to 5 divided by log 10 to the power 6 divided by 10 to the power 3, that is equal to 5 log base 10 to the power 3, that is 5 by 3, that is equal to 1.67 minutes. This is the answer. Now, half-life period. It is the time required to reduce the microorganisms concentration by half. That means, if n0 by n is equal to 2, then T is equal to T half. So, from equation 3, n by n0 is equal to e to the power minus kt, that means T is equal to ln n0 by n divided by k. So, T half is equal to ln 2 by k, then T half is equal to 0.693 divided by k. The thermal death time, that is F value. The thermal death time is the amount of time that is necessary to kill a specific number of microbes at a specific temperature. This value is obtained by keeping temperature constant and measuring the time necessary to kill the number of cells specified. This time is expressed as a multiple of d value as long as the survival curve follows a first order model. From example, log base 10 n0 by n is equal to T by d. The T is equal to F is equal to d log 10 n0 by n. As an example, if we have 100 microorganisms and we want only 10 survivors, then F is equal to d into log 100 minus log 10, that is 1d. In fact, in going from 100 to 10 microorganisms, we achieve a 90% reduction, and thus F value is equal to d. Here follows other examples for 99% reduction and 1 survivor. F is equal to d into log 100 minus log 1, that is 2d. For 99.9% reduction, 0.1 survivors, F is equal to d into log 100 minus log 0.1, that is d 2 minus 1, that is 3d. Likewise, for 99.99 reduction, that is 0.01 survivor, F is equal to d into log 100 minus log 0.01, that is d 2 minus 2, that is 4d. So, another example let us take, that the d value of a spore is 2 minutes at 121 degrees Celsius. So, how long is needed to achieve a 12 log reduction? So, here we know that F is equal to d into log reduction, that is equal to d, that is d is 2 minutes, 2 into 12, that is 24 minutes. Another example let us take, a thermal process follows a d value of 3 minutes at 121 degrees Celsius with a z value of 10 degrees Celsius. The process is conducted at 110 degrees Celsius for 15 minutes calculate the F value. So, as we know that F is equal to T into 10 to the power T minus T reference temperature divided by Z. So, that is equal to 15 into 10 to the power 110 minus reference temperature, that is 121 degree Celsius, divided by 10, that is equal to 15 into 10 to the power 1.1, that is 15 into 0.079, that is 1.185 minute. That is the answer. In another example, a food product is treated at 121 degrees Celsius for 10 minutes with a D value of 2 minutes. At this temperature, how many log reductions occur? So, log reduction is equal to F by D, that is 10 by 2, that is 5 log reduction. Now, 12D concept. Canned foods are susceptible to the spores of the organism Clostridium botulinum. This is the organism that causes botulism. These bacterial spores can survive many heat treatment processes. However, in modern food production, canned foods are subjected to a time temperature process that will reduce the probability of the survival by the most heat resistant Clostridium botulinum spores by 12 logs or 12D at 121.1 degrees Celsius. The temperature used in the calculation of most commercial 12D processes is the 121.1 degrees Celsius, and the D value for this organism at 121.1 degrees Celsius is 0.21 minutes. For canned foods, Clostridium botulinum is the primary organism of concern, and it has been established that the minimum heating process should reduce the number of the spores by a factor of 10 to the power minus 12. This means that since D is the time required to reduce the original number by 10 to the power minus 1, the substituting n by n0 equal to 10 to the power minus 12 into equation 6, we get T is equal to D into log base 10 n0 by n, that is T is equal to D into log base 10 to the power 12, because n by n0 is equal to 10 to the power 12 minus 12. So, n0 by n, just inverse of that is 10 to the power 12. So, T is equal to 12D, where D is equal to 2.303 by K. This is often called the 12D concept. Now, temperature dependence of reaction rates. The Arrhenius equation. The activated complex theory for chemical reaction rates is the basis for the Arrhenius equation, which relates reaction rate constants to the absolute temperature. The Arrhenius equation is K is equal to A0 e to the power minus EA divided by RT, where EA is the activation energy and A0 is the rate constant as T approaches infinity. Another form of the Arrhenius equation involves the reaction rate constant at a reference temperature. Let T0 is the reference temperature at which K is equal to K0. So, K is equal to A0 e to the power minus EA divided by RT, this is the equation 10, and K0 is A0 e to the power minus EA divided by RT0, that is equation 10. So, let us take the ratio of equation 10 and 11, that will reduce to K by K0 is equal to e to the power minus EA divided by R into 1 by T minus 1 by T0. Further, since D is proportional to 1 by K, so this will reduce to D0 by D1, that is equal to K by K0, that is equal to e to the power minus EA divided by R into 1 by T minus 1 by T0. The negative sign is placed on the exponent of the Arrhenius equation in order that a positive activation energy will indicate an increasing reaction rate constant with increasing temperature. Using equation 11, the rate constant at any temperature can be determined from the activation energy and the rate constant K0 at a reference temperature T0. Now, Q10 value. The Q10 value of a reaction is often used for reporting temperature dependence of biological reactions. It is defined as the number of times a reaction rate changes with a 10 degrees Celsius change in temperature. If a reaction rate doubles with the 10 degrees Celsius change in temperature, the Q10 is equal to 2. Thus, the general rule of thumb in food storage is that a 10 degree reduction in storage temperature will increase shelf life by a factor of 2. The relationship between Q10 value and the activation energy is derived as follows. Let K1 is the rate constant at temperature T1 and K2 is the rate constant at temperature T2. So, K2 is equal to K1 Q10 at T to the power T2 minus T1 by 10. Let us take the natural log of this, then natural log of K2 divided by K1 is equal to T2 minus T1 divided by 10 natural log of Q10. This is then from equation 11 we can write that K2 by K1 is equal to e to the power minus EA divided by R into 1 by T2 minus 1 by T1. Taking log of equation 15, which will be reduced to natural log of K2 by K1 is equal to minus EQ divided by RT, T1 minus T2 divided by T2 into T1. The negative sign of EA in equation 16 drops out when the sign on T1 and T2 in the numerator are reversed. Equating equation 14 and 16 and solving for EA by R. So, E minus EA by R is equal to natural log of Q10 by 10 into T2 into T1, or Q10 is equal to e to the power EA by R into 10 by T2 into T1. The Q10 is temperature dependent and should not be used over a wide range of temperature. Now, thermal resistance constant, that is Z value. The Z value is the number of degrees of temperature necessary to reduce the D value by 1 log cycle, that is to kill 90% of the population of a microorganism. For Z values, temperature is the variable with time being constant. At the It is the reciprocal of the slope resulting from the plot of the logarithm of the d value versus the temperature at which the d value was obtained. While the d value gives the time needed at a certain temperature to kill an organism, the z value relates the resistance of an organism to differing temperature. The z value calculates a thermal process of equivalency if given one d value and the z value. This is the pictorial representation of z value. Here if the log thermal death time is plotted against the temperature, then this 1 log cycle reduction is called the z value. This is the same representation, that d value is plotted, that is the thermal death time versus the temperature, and this is the z value for 1 log cycle reduction. The traditional approach to thermal processing has used the term thermal resistance constant, that is Z, to describe the influence of temperature on decimal reduction time for microbial population. Thermal resistance constant is defined as the increase in temperature necessary to cause a 90% reduction in decimal reduction time, that is d value, that is z is equal to T2 minus T1 divided by log T dt1 minus log T dt2. Letting time that T1 is equal to 121.1 degrees Celsius, which is the standard temperature against which thermal process are compared, and calling T2, that is T, in equation 14, that will become dt is equal to d 121.1 into 10 to the power 121.1 minus T by Z. For Clostridium botulinum, Z is equal to 10 degrees Celsius. This means that increase in temperature of 10 degrees Celsius will increase the death rate by a factor of 10. Using equation 6, T is equal to dt into log base 10 n0 by n. Now substituting T is equal to 121.1 degrees Celsius as the standard temperature into this equation, and substituting F0 for T, F0 becomes d 121.1 log base 10 n0 by n, where the F0 value of the process is the time t in minute at 121.1 degrees Celsius, that will produce the same degree of sterilization as the given process at its temperature T. The F0 of the given process at temperature T is now given as F0 is equal to d 121.1 into 10 to the power T minus 121.1 divided by Z. Now these are the some examples for you to solve based on the formulas we have discussed so far.